This diagram represents the foundational rule of modern biology, known as the Central Dogma. It illustrates a strict, linear flow of genetic information, starting at DNA, passing through an RNA messenger, and resulting in a functional protein. Francis Crick articulated this directional sequence in 1958 of messenger, and proteins function as the final hardware. While this linear simplicity became the cornerstone of molecular biology, it created an illusion of order that obscured the chaotic reality of the genome. Researchers eventually uncovered biological mechanisms that directly collided with this textbook model, identifying viruses that drive genetic data in reverse, genes that slice themselves out of chromosomes to move to new locations, and infectious proteins that spread disease without using any DNA. These discoveries raised a difficult question. Had these anomalies invalidated Crick's directional rules entirely? Scientists now recognize life's source code as a dynamic network of interacting molecules, moving past the idea of a simple read-only archive. In 1970, the discovery of retroviruses and an enzyme called reverse transcriptase triggered a major reassessment of genetic flow. This enzyme allows a viral RNA genome to be copied backward, creating a new molecule of double-stranded DNA. This newly minted viral DNA then travels into the nucleus to splice itself directly into the host organism's own genome. By receiving and permanently storing data generated from an RNA template, the host DNA proved that genetic information can flow in both directions. Geneticist Barbara McClintock challenged another core assumption, the belief that genes occupy fixed, stable positions on a chromosome. These maize kernels provide physical evidence of her discovery. Their patchy coloration is the direct result of genetic disruption during development. This disruption is caused by jumping genes, DNA sequences that actively cut and paste themselves into new locations, altering how the surrounding genes are expressed. The RNA world hypothesis identifies RNA as an active catalyst and master regulator, performing complex cellular tasks independently of DNA. Thousands of non-coding RNAs function like traffic cops, intercepting genetic messages to silence or amplify specific instructions before they are ever translated. The genome functions as a volatile, self-editing architecture, where genetic instructions are constantly reshaped by RNA networks. Prions represent the most extreme departure from the dogma. They are infectious agents composed entirely of misfolded protein, containing zero DNA or RNA. They propagate through a physical mechanism where a misfolded protein forces normal proteins to misfold, transferring structural information without a genetic template. This dominance of physical form is also evident in epigenetics, a regulatory layer where the underlying sequence of letters remains intact while the cell determines which genes are actually utilized. Chemical markers like DNA methylation physically attach to the genome to block access to specific genes, preventing them from being read. The DNA molecule itself can contort into alternative shapes, such as the left-handed zigzag of zDNA, to regulate how the code is transcribed. Biological information relies on an interactive overlay of 3D shapes and physical modifications that control the flow of data from the DNA. Scientists eventually had to determine if prions, jumping genes, and reverse transcription had truly invalidated Crick's original directional rules. These anomalies actually depend on the rules they appear to break. A prion, for instance, requires the host's standard DNA to manufacture the original protein before the misfolding process can begin. Reverse transcription was eventually accommodated within Crick's own framework when he revised the model in 1970. Crick's directional rules provide the stable archive for a system that filters, amplifies, and redirects its own output through layers of physical control. This glowing network reveals an interconnected map. DNA operates as a central node, with RNA and proteins forming complex orbiting feedback loops. Mechanisms like epigenetics and jumping genes dictate exactly when, where, and how loudly the existing code is read, without altering the underlying sequence. The genome operates as a responsive machine, altering its own internal state to meet the shifting demands of survival. To continue exploring the mechanisms of the genome, please like this video and subscribe to Computational Genome Biology.